This is Rajik out here, and I welcome you to my channel Learn and Smile. Previously in this channel, we have analyzed and discussed about the DNA replication process taking place in the prokaryotes. And while doing so, we have also discussed the different types of DNA polymerases that do play a very active role in the same process. Now we started with DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4 and DNA polymerase 5, where I have also added that DNA polymerase 3, known as the DNA replicase, is the most prominent enzyme that do play a very pivotal role in the DNA replication process of the prokaryotes. Now after that, we have tried analyzing the DNA replication process in the prokaryotes with the help of an example, that is through E. coli, where we have understood that E. coli do possess a single chromosomal DNA, and we have tried to analyze different prominent structures of that particular chromosomal DNA of the E. coli, where we have seen that there is an origin of replication which is known as the ORIC, and within the ORIC there are certain repetitive structures ranging from 9' sequences, 13' sequences, and then 5' GATC sequences, that is 5' to 3' GATC sequences, and what the importance of these. Now you guys, if you have not yet checked it out, then please do check in my previous videos and then come to this particular video, because do remember one thing: there is an English saying that everything that glitters is not gold. The reverse is also true, that everything that doesn't glitter doesn't mean that it is a crap. So my previous videos are in sort of black and white, but it doesn't mean that it will not help you out, because the videos that I'm talking about are the building blocks for this particular current videos. So if you have not checked it out, please go and check it out first and then come back to this video, where we are going to analyze the entire story of DNA replication taking place in the prokaryotes. Hope you will love this particular video, and thank you very much for having this ample amount of patience with me. So without any further delay, let's begin. So let's conclude the very beautiful journey of analyzing the DNA replication process taking place in the prokaryotes. And we'll conclude this journey by fragmenting our discussion into 3 separate modules. In the first module, we will be discussing about the initiation of DNA replication. In the second module, we will be discussing about the elongation of the DNA replication process. And in the last module, we will be discussing about the termination of the DNA replication process. Starting with the first module, that is initiation, the very first thing we need to focus on is the ORIC, that is the origin of replication. Now, the origin of replication is 245 base pairs approximately. Now, this ORIC, it envelopes certain repetitive sequences which is AT rich. Now, these repetitive sequences are present in different numbers. Say for example, there are 5 units of 9' sequences, 3 units of 13' sequences, and 11 units of GATC sequences aligned in 5' to 3' direction. The significance of all these 3 sequences have been already discussed in my previous videos. Now, come to this particular structure. Assume that this segment or this region of the DNA which I have drawn is the ORIC. This ORIC envelopes the repetitive sequences which is AT rich, that is, it envelopes the 13' sequences, 9' sequences, and GATC sequences which is aligned from 5' to 3' direction. Now, starting with the DNA replication process, this 9' sequences puts the first step, because this 9' sequences provides the binding site for a very special protein known as the DNA protein. Now, this DNA protein itself has got 2 binding domains. With the first binding domain, it, for the first and foremost time, it binds with the 9' sequences. Now, after binding with the 9' sequences with the other domain, if bounded to the ATP molecule, then it can interact with the 13' sequences as well. As the interaction with the 13' sequences is established, it will create a strain in this particular region, and eventually it will melt this region, producing an open complex. Now, with the formation of the open complex, there is an invitation for the other replication enzyme to come in and join their hands in the process. But before that, you need to understand that at the same time there is methylation at the GATC sequence, where methylation in the adenine residue of the GATC sequence demarcates the regulation process of the DNA replication, which again I have discussed in my previous video. Okay, having understood this, that is, having this sequence methylated and opening of the 13 merge sequences, forming the open complex and providing an open invitation for the other enzymes to join in, the helicase comes in as the second enzyme. This helicase, it binds to the lagging template strand, which is in the 5' to 3' direction. Now it is to note that it is DNA B which acts as the helicase for the prokaryotes. Now this helicase, it binds to the lagging template strand with the help of DNA C, which is the helicase loader. Basically, this lagging template strand is the parental strand which will act as a template strand for the future lagging strand. And that lagging template strand must be in the direction of 5' to 3', because it is when the lagging template strand is in the direction of 5' to 3', it is only then the helicase binds in. And after binding in, the helicase melts the double-stranded DNA. With the melting of the double-stranded DNA, there comes the DNA polymerase 3. But the DNA polymerase 3's arrival is not that a happy one. Why? Because as the DNA polymerase comes in, it is very sad. Why? Because there is no 3' OH, and we know that without the 3' OH, this DNA polymerase 3 has got no job. So since it requires the 3' OH, and it recruits another enzyme known as the primase. Hope it is visible, right? Okay, so this DNA polymerase 3 recruits another enzyme known as the primase, which is DNAG in case of prokaryotes. It is this primase which synthesizes the primer. Basically, the primer is a short stretch of RNA molecule which is synthesized in order to produce the 3' OH end to the DNA polymerase, so that the DNA polymerase, after acquiring the 3' OH end, can conclude its functions. Alright. Now with the synthesis of the primer, see, the DNA polymerase 3 is very happy, and then it starts nucleotides addition in the form of DNTP. Now DNTP stands for deoxyform of nucleotidyl triphosphate, right? It is the deoxyform of nucleotidyl triphosphate. It might be deoxyadenosine triphosphate, it might be deoxycytosine triphosphate, it might be the deoxy form of guanosine triphosphate, and so on. Now, after the DNA polymerase starts the polymerization activity, that is addition of the nucleotides. Now, as already discussed, that as the helicase or the replication form moves forward, replication also proceeds forward. As the replication proceeds forward, this helicase also proceeds forward. Now, with the forward direction of this helicase, there is a tendency for the previously melted DNA to re-anneal, and here to prohibit it, there comes another protein which is known as the Ssb protein, which stands for single stranded binding protein, which inhibits the re-annealing. And also with the movement of the DNA helicase along with the replication fork, there is a positive supercoiling towards the moving end. And in order to remove the positive supercoiling, there comes another enzyme which is known as the DNA guidase. Now it is the DNA guidase that removes the supercoiling. Basically, this DNA guidase is one of the example of topoisomerase, which plays a very pivotal role in removing the supercoiling in the prokaryotes. So if I have to sum it up, then I would say that first is the 9 merge sequences to which the DNA A protein binds. Then comes the interaction of the DNA A protein with the 13 merge sequences. But DNA A protein can interact with the 13-mer sequences only when ATP is binded to it. After interacting with the 13-mer sequences, an open complex is formed. Along with the formation of the open complex, it is very important that methylation of the adenine residue present in the GATC sequences is achieved. Now, with all these prerequisites done, there comes other enzyme known as helicase, which is responsible for the melting of the double-stranded DNA. With the double-stranded DNA being melted, there comes DNA polymerase. But since DNA polymerase doesn't find the 3' OH, it recruits another enzyme known as the primase. The primase forms the primer which provides the 3' OH to the DNA polymerase 3. Now the DNA polymerase 3 with the help of 3' OH starts the nucleotides addition, that is polymerization, thereby elongating the newly synthesized strand of DNA. Now with the replication process moving forward, there happens two events. One, that is there is a tendency for re-annealing of the previously melted DNA. For that, there is another protein which is known as Ssb protein which help inhibit the re-annealing. And the second event which occurs is because of the forward movement of the replication fork, there is supercoiling that is being developed, and in order to remove that supercoiling, there is another enzyme, that is topoisomerase, which is DNA guidance in case of prokaryotes, comes in and help remove the supercoiling. So this was the entire initiation process. So having understood the initiation process, let's move on to the elongation process. Now come to the elongation process. We know that the DNA polymerase 3 requires a 3' OH end, which is provided by an enzyme known as the primase, which is DNA-G in case of prokaryotes. This primase, with the help of the synthesis of a primer, provides 3' OH to the DNA polymerase. Now, one thing you need to understand that within the DNA polymerase, there is a presence of two divalent metal ions. Now, these two divalent metal ions may be either manganese or zinc, but these metal ions are present in two units. One of the unit of the metal ion interacts with the H atom of the OH end, and after interaction it provides an alkoxide ion. This alkoxide ion, after getting converted into an attacking group, attacks the incoming molecule, which is ATP. Now as you can see that the bond where the first phosphate is associated with the second phosphate is known as the alpha bond. The bond where the second phosphate is associated with the third phosphate is known as the beta bond, and the last bond is known as the gamma bond. This alkoxide ion attacks this alpha bond and it breaks it into two different molecules. The first molecule is the AMP, which gets associated with the alkoxide ion in this particular way, and the second is the pyrophosphate, which thereafter is hydrolyzed with the help of an enzyme known as phosphatase. Now with the addition of AMP to the alkoxide portion of the primer, we have another 3' OH end. Now with similar interaction, this 3' OH end will also gain another nucleotide which would be complementary to the parental strand, and in this way the polymerization activity will move ahead. Now as the process comes to an end, the primer which is incorporated needs to be removed, and for the removal process DNA polymerase 1 comes into action. As we know, the DNA polymerase 1 has got 5' to 3' exonuclease activity, which is very much efficient to remove the primer. Therefore DNA polymerase 1 will remove the primer. Now after the removal of the primer, there are gaps created. Now this sealing job, that is the sealing of the gap, will be eventually done by DNA polymerase 3, which is followed by the ligase. Now if you guys want to know that how does ligase play a role in the sealing of the gap, then do comment in the comment box below. I will provide another video where I would explain how the enzyme ligase help in the sealing, in the complete sealing rather, of the newly synthesized strand. Now since this video is getting longer, so I have decided to stop our journey till elongation. We would be discussing and analyzing the last module, that is the termination of the DNA replication process, in our next video. Till then, stay tuned.